Hello, thank you for being here with me today and welcome. This talk focuses on the effects of genetic changes in the insulin-like growth factor and growth hormone families on growth, metabolism, and endocrine regulation. I'll start today by giving a quick rundown of the signaling pathway for growth hormone and insulin-like growth factor and how it affects endocrine regulation. After that, I'll go over the study's experimental setup and the main conclusions drawn from changes in the mice growth hormone and insulin-like growth factor genes. Lastly, we'll look at the implication of these findings for the hormonal control of metabolism and growth. Let's first look at the overall picture of how growth hormone and insulin-like growth factor normally function in the body. I will refer to them as GH and IGF-1 for short. The anterior pituitary secretes growth hormone, which enters the bloodstream and makes the way to the liver and other target tissues. GH promotes the synthesis of IGF-1 after attaching to the liver cell receptors. Many of the GH's effects, such as bone elongation, tissue growth, and cell proliferation. So they're primarily mediated by the IGF-1, and it acts locally at tissue sites and circulates in the blood system. It's critical to realize that GH and IGF-1 are components of a strictly controlled feedback loop, which is inhibiting GH secretion, which is elevated at the IGF-1 levels, aid and avoiding overstimulation. This equilibrium is essential to this. So we will also do the study that we will discuss also tested what happens when the system is genetically disrupted in different ways. So the researchers developed a variety of genetically modified mouse models to further study this axis. This comprised transgenic models which overexpress particular genes and knockouts in which particular genes were rendered inactive. So this created, for instance, mice with liver-specific IGF-1 deletion, which means they didn't have the gene. And without the GH gene, mice without the GH receptor. They also produce mice with excessive IGF-1 or GH. Because of this, they were able to examine both extremes of the signaling spectrum. So the consequences of excessive and insufficient hormone signaling. So crucially, these models assisted in differentiating between the functions of locally and systematically produced IGF-1, which has proven difficult to investigate in living things, which is good. So in order to look in further, let's take a look here in the further of this image. So let's further examine the picture and summary of transgenic knockout mouse lines with different GH and IGF action in more detail. So let's look at the picture more closely. So proteins involved in GH secretion, regulation, GH-induced intracellular signaling, and IGF-1, ALS, and IGF-BP3 production are all depicted in the diagram. The various mouse colors you see here on the right-hand corner, those stand for transgenic overexpressed mice, which are black. Mice with globally knocked out genes are white, adult onset knockouts are blue, or tissue-specific knockouts are purple. So this kind of like helped out setting it up and kind of like in an axis what they're trying to explain. Over here you see the anteropituitary gland, which is showing you how it works with the different mice. Let's go over the findings. Researchers observed significant alterations in growth and metabolism in the mice that either had the GH gene or the GH receptor that had been deleted. So these mice were noticeably smaller than usual, which should come as no surprise. Their bones were shorter and their body weights were lower. So interesting enough, though this knockouts also displayed metabolic benefits, although their growth was stunted, they had improved glucose tolerance and increased insulin sensitivity. So, moreover, these mice outlived the control group, which reduced the GH signaling that may help people live longer because what it does, it reduces stress and metabolic demand. So the research demonstrates that GH's growth promoting effects are mostly indirect. This means that it acts by inducing IGF-1 production rather than directly influencing the tissues. Let's take a closer look at this image that we have here. So it's comparing the phenotypes of multiple transgenic and knockout mouse lines with different GH action. So the mice under comparison is their relative size and the genes that have been altered. So are being shown on the top figure. So if you do look on the top, you do see them differently. And then there's a comparison to WT controls. So the red box denotes higher growth and the blue box denotes lower growth. And the gray box shows no change at all in growth. So now let's examine the results when you're knocking out the GIF1 gene. So growth was significantly kind of disrupted in the mice with complete knockout. So in which IGF-1 was removed from every tissue, given how crucial it is for development, these mice were even smaller than the GH knockouts. So on the other hand, growth was considerably less effective when IGF-1 was only deleted in the liver. So this implies that GIF-1 generated in other tissues such as muscle, fat, bone continue to be important. The fact that serum IGF-1 levels only decreased the complete knockouts and not in liver-specific ones was particularly intriguing. But, however, growth was still hindered in both, demonstrating the significance of IGF-1's local actions in addition to the circulating levels. So if we were to put it another way, although the majority of IGF-1 is in the blood that comes from the liver, it is not the only type of IGF-1 that is important for actual tissue growth. So on the other end of the spectrum, when GH is overexpressed in mice, they developed a condition similar to organomegaly, a human disorder characterized by excessive GH. These mice were larger than normal with heavier organs and larger bones. But the downside was significant. They developed insulin resistance, elevated insulin levels, and reduced lifespans. So the overproduction of GH also caused high circulating IGF-1, which likely contribute to the excessive tissue growth. So this experiment illustrates the risk of hormonal excess with more GH and can boost growth in the short term, and it comes to the cost of metabolic health and longevity. The study also found that mice with increased IGF-1 levels had a milder phenotype than those overexpressing GH. They were slightly larger but did not experience metabolic issues or organ overgrowth. Their lifespan was slightly shorter than normal, but overall they were closer to normal. So the results supported that the idea that IGF-1 has a more regulated tissue-specific function in growth with less severe effects compared to GH. So in summary, the study validates a number of key endocrine biology ideas. First of all, rather than directly affecting tissues, growth hormone merely uses IGF-1 to indirectly promote growth. Secondly, circulating IGF-1 from the liver is not as important as IGF-1 produced within tissues, if not so more. And then third, while both hormone excess and deficiency cause health issues, they do so in quite different ways. Growth is stunted by insufficient GH and our IGF-1, whereas excessive results in insulin-resistant metabolic diseases and shorter life expectancies. So these discoveries have significant ramifications for the management of growth disorders and our comprehension of how the endocrine system strikes a balance between growth and long-term health. So here, let's look up the closer image where it's the GH and IGF family of mouse lines that has grown significantly since 1980 and the first tissue-specific line created in 1999. So current research focused on 137 mouse lines, each advancing our understanding of the effects of GH and IGF-1 on health and disease using transgenic and knockout technologies. So thank you so much for joining me today. I'm hoping you really liked the discussion. Thank you.